Because the digestive system has the largest number of different kinds of organs, it might be of value to quickly review each major component along with its primary function. The mouth and associated structures, including the gullet and pharynx, are involved in ingestion, mastication, and swallowing of nutrients. The esophagus transports ingesta to the stomach. The stomach temporarily stores ingesta and begins hydrolysis of proteins. The small intestines is the primary organ of hydrolysis and absorption. The large intestine absorbs water and electrolytes to dehydrate remaining ingesta and form fecal material. The salivary glands secrete a lubricating fluid to facilitate swallowing. The pancreas secretes a powerful digestive fluid containing several different hydrolytic enzymes. The liver metabolically modifies and detoxifies material absorbed from the alimentary canal. The gallbladder serves as a temporary storage vesicle for bile produced by the liver.
Hi, I'm Mike Shively. Welcome to Introduction to Anatomy and Physiology. We're talking about the digestive system. Any of you have any questions from our last session? Starley?
I know that in the large intestine you said that they were colic flexures. What was a flexure?
A flexure in the intestinal tube or tract is just a bend. And you know, let's go back to that large intestine. Let's go to the overhead camera for just a second. And let's kind of draw the large intestine here. Comes across, comes down, has a little bit of a curve here. Not quite that much of a curve, but. And if this area right here is where the ileum comes in, then we said that below that level is cecum. This is the cecum, and the vermiform appendix projecting off that is a finger-like projection. Coming up here would be the ascending colon, and this bend is called the right colic flexure. So that's a flexure because it's a bend. This bend is called the left colic flexure. This kind of an S-shaped curve, which is quite subtle down here, is called the sigmoid colon, but it's a flexure as well. And then another thing that nobody's asked about, but the large intestine has kind of a pleated appearance as opposed to the small intestine, which does not have this pleated appearance. Anybody know what causes that, the pleats, or the sacculations, or hostra they're called? Eva?
It's caused by the tanii coli, which is kind of a band of smooth muscle.
Right. When we looked very quickly at the layers of the intestinal wall last time, there's the smooth muscle tissue on the outside called the tunica muscularis in the large intestine has some longitudinally oriented bands in it that are called tenia coli. And those longitudinally oriented bands, if we go back to this overhead camera, tend to contract a little bit and pull the large intestine up into these, most of the large intestine, certainly the cecum and colon, which is most of it, up into these pleats, or the little bulges are called sacculations or hostra. At any rate, Starley, those bends are called flexures. And one of the other things that we didn't look at, really, it's kind of intriguing when you're comparing large intestine to small intestine in a gross lab. Not only do you have the length difference, where the small intestine is so much longer, 21 feet versus 5 or 6 feet for large intestine, you also have villi present in small intestine and not present in large intestine. But you have these sacculations that are really easy to see from clear across the room. Matter of fact, let me pull this model back up. Let's look at it on the overhead camera for just a second. Got more than I bargained for here, I think. But in the gray, we have a large intestine here. And these are the pleats that I'm talking about. Each one of these is a sacculation. And what Eva mentioned are these longitudinal bands of smooth muscle tissue. There's actually about 3 of them as we go around the wall of the intestine, although only one will present itself to us as we look at it from this particular perspective. And the small intestine does not have these individual bands of longitudinally oriented smooth muscle, and as a consequence, the wall of the small intestine is not pulled up into those pleats. Adam?
What's the purpose of those sacculations?
I think it's just the normal orientation having to do with the way peristalsis occurs through the large intestine. Remember, peristalsis is the name we give to the progressive squeezing that the smooth muscle tissue in the wall of the intestinal tube undergoes in order to move ingesta, through the alimentary canal. Of course, every once in a while you get something down in your stomach that doesn't agree with you to the point that you do the little reverse peristalsis thing and empty out the stomach and by vomiting. That's actually primarily a contraction of the stomach wall itself, which is just simply forcing the ingesta up through a dilated esophagus. Not particularly a pleasant experience, although those of us who have gone through it know that you feel a bit better after that occurs than you do before, because usually you're pretty severely nauseous before you vomit. Bryce?
There's quite a few people that have had the vermiform appendix removed. Does that have any major roles that it plays?
Functions of the vermiform appendix in humans are basically negligible. But in some mammals, the appendix is a functional organ. Rabbits have a functional appendix, for example. It's used for the microbiological breakdown of fibrous materials, celluloses that you and I can't ingest at all. Bacteria can have the enzymes necessary to break down some of those, and some of that bacterial degradation does occur in the appendix. Most mammals have a functional cecum, which is that initial part of the large intestine, but most mammals, the appendix is not functional. It's a vestigial remnant. Going back to your question, though, Starley, some other flexures that we didn't talk about last time. I want to go to the overhead camera for just a minute, and I want to redraw the stomach here. I'm going to start up here at the top and make the stomach kind of small because I want to spend most of the time on the duodenum of the small intestine. So the duodenum comes down like this. It actually bends, slants up like this. And so we're going to say from right here to right here is duodenum. Now that total distance, and let's label this stomach, okay? Remind ourselves what that is. So the esophagus is coming in here, attaching at this point, and the duodenum itself is divided into 7 or 8 named segments, even though it's only 1 foot long in total. This initial part of the duodenum, which is still kind of oriented in a superior direction, if you will, is called the superior part of the duodenum. And this, Starley, is the superior duodenal flexure, that bend, that umbrella handle bend. So we got the superior part of the duodenum, the superior duodenal flexure. This is the descending part of the duodenum because ingesta would be moving downward as it passed through that part of the lumen. This bend down here is called the inferior duodenal flexure. This is the horizontal part of the duodenum. It slowly slants up as the ascending part of the duodenum. And this bend right here, which is about a 180-degree turn, is the duodenal jejunal flexure. So we got superior duodenal flexure, we've got inferior duodenal flexure, and we've got duodenojejunal flexure. All of those bends, named bends, and several straight parts named within, 1 foot segment. And what's really intriguing about this from an anatomic standpoint is you go to the ileum and the jejunum next, and then the ileum. There are essentially no named landmarks, even though we've got several feet of ileum and jejunum. Any other questions? Amber.
This is actually a question about snakes. Like when they eat rats or whatever, do they have stomachs? Because you can see when they eat it, it moving through their body.
They're able to, a lot of snakes are able to ingest very large objects, much larger than the biggest part of the snake would be in transverse section. They have a kind of an interesting jaw structure where there's a quadrate bone and they can split their jaw on the mandibular symphysis apart sideways as well. So they can really get a huge opening here to where they can take in something quite a bit larger than their own diameter. And then, as you mentioned, as the snake continues to swallow, that whatever it is, you can follow it down, and there'll be a lump there. Now that goes down into the stomach region where powerful digestive enzymes and so forth break it down, and it'll get smaller over the next several days. But depending on the size of the snake and what's been ingested, they can go for quite some time in between meals. But yeah, the material is in the stomach, and then it does move on down into lower parts of the alimentary canal. Any other questions? Eva?
In some of the sections of tissue from the small intestine and other stuff that you showed us last time, there were different layers of muscle tissue, smooth muscle tissue, that were going in different directions. The fibers were running in different directions. Why? What's the point of that? Why do they go in different ways?
I think orientation of the smooth muscle fibers, in particular in two major planes in most of the alimentary canal, somehow facilitates that peristaltic process or that squeezing process. Actually, when you get into the stomach wall, there are three distinct layers of smooth muscle tissue oriented in different directions, like the plies of plywood, which make the stomach very efficient in that the squeezing that occurs in the emulsification or mixing of ingesta with gastric secretions. I know that's not a specific answer, but that's the general idea. Well, let's talk a little bit about the digestive glands, if you will, some of the organs that actually produce secretions that are carried by duct systems into the alimentary canal. And we'll start with salivary glands. Now, the salivary glands produce a fluid called saliva, and a lot of people think that there's a lot of important digestive functions to the saliva. Well, it's not probably as much as we would ascribe. Saliva is mainly used as a lubricant for the swallowing process. Have you ever been in one of those, let's eat a bunch of saltine crackers and see who can whistle first contests? I don't know what you do at birthday parties in this state, but anyway, you know how important it is for swallowing to have something. Matter of fact, a lot of times with meals, in order to make the swallowing process easier, we have some water or other liquid that we imbibe along with the food that we're taking in. The main purpose of saliva is in the swallowing process to lubricate the ingesta. There is one weak digestive enzyme that's found in saliva. Does anybody know what that enzyme is? Adam? Amylase. And amylase breaks down what? Starch. Let me write that out for everybody if we go to the overhead camera. Amylase is a hydrolytic enzyme that breaks down starch, and of course it breaks down starch into glucose. You can experience this yourself. You put a piece of bread in your mouth and start chewing on that bread. Bread doesn't taste particularly sweet when you first put it in, but you keep chewing on it way past when you normally would have swallowed it, and you'll notice it begins to taste a little bit sweet because the amylase that's present in the saliva is breaking this starch down into simple sugar, glucose. And so when we think of salivary glands, I want you to think primarily of lubrication. Now, we've got three major pairs of salivary glands. Let's go to a graphic that summarizes what the names of those are. Left and right parotid glands, left and right sublingual glands, and left and right submandibular glands. These are the official names. Quite often people will stick the word salivary in there, so they'll call it the parotid salivary gland or the sublingual salivary gland. Now, there are other salivary glands besides these three major pairs. There are real small ones, kind of microscopic ones that are present on the mucosal surfaces of the cheeks and lips, and some that are present on the surface of the tongue as well. But those are the three major pairs. And I want to show you those on some models just so you can see the kind of location. If we go to the overhead camera, here's a head model, and this structure right here is the parotid gland. Now, PAR comes from a Latin word root that means near, and OT, it means ear in Latin. So this is the near the ear gland, if you will, and you can see that it is located very near the ear. If you look carefully, let me get a pointer here, These yellow things here are nerves. But this slightly discolored yellow structure going across the surface of the masseter muscle is the parotid duct. And it carries the secretion from the parotid gland through the cheek into the oral vestibule right next to some upper teeth. And so that's how the saliva from this particular gland gets into the oral cavity. In addition to the parotid gland, we've got the sublingual and submandibular glands that are located down here. Now this model just shows one structure. I want to bring up a big model here that we've looked at previously because on this particular model you can see the sublingual and submandibular glands are located right adjacent to each other, and typically the major portions of those share a common duct which actually opens at a sublingual papilla that's located right here. It's actually called the sublingual caruncle. It's a little papillary projection. Now, have you had the experience ever of maybe just yawning or something, kind of minding your own business, and all of a sudden you do the little spitting cobra routine? Have you had that particular experience? I learned when I came to Utah that y'all have a word for that, and it's called gleeking here. I don't know if that's, I haven't looked up gleeking in the medical dictionary, but basically what's happening in that instance is during that yawning process, some very thin muscle tissue under here is pressing on the sublingual and submandibular glands and actually just squeezing saliva out through this particular duct. And typically in a yawn, you kind of roll your tongue back and and open your mouth, you know, and so the saliva will just make an unwanted and unwelcome appearance outside your body, particularly for somebody sitting right in front of you. It's unwanted and unwelcome. At any rate, that's what that's all about. So those glands then produce this saliva, which is used to lubricate ingesta for the swallowing process. And the duct from those two actually empty underneath the tongue. So we've got parotid gland that opens in the oral vestibule, that's between teeth and cheek, and then the other two pairs open underneath the tongue on sublingual caruncles. Any questions about salivary glands? There is a little bit of difference in the particular character of the secretion from one salivary gland to the next, but it's not a significant one. I think maybe I may have mentioned when we were talking about the eye, sometimes the lacrimal gland, which produces tears or lacrimal secretion, in some cases that lacrimal gland doesn't produce adequate secretion, and so you have a dry eye syndrome, which can be uncomfortable to downright painful. And so your alternatives are to perhaps instill artificial tears into that eye several times a day, which you can buy across the counter at a drugstore, or you can opt to have a parotid duct translocation, where an ophthalmic surgeon can move the duct of the parotid gland from the oral cavity exit or position up into the conjunctival sac and moisturize your eye in that manner. Eva?
This is a little bit of a side topic, but I've noticed when I eat something that's really sour, or if I think about eating something that's really sour, I'll get this pain right here, about where you said the parotid gland was located. Does that have anything to do with the parotid gland, or is that just me? I don't know if that happens to everyone else.
It's not just you, because I have that experience myself, and I can't explain it, but if it's really sour, or I know it's going to be really sour, I get that back in here myself, yeah. And I can't explain that. Does anybody have an explanation for that? Any other questions or comments? Does anybody have a question that I can answer? Maybe not. Okay. Let's move on down to, let's go to the pancreas next. And I want to go to the pancreas because it's a relatively simple organ. If we go to the overhead camera, if I were to draw a pancreas, I can just about draw any shape I want here. That's kind of about the way one looks, and this is the head of the pancreas. This part's called the body. It's not considered to have a neck, and this part's called the tail. And this sits right next to the stomach. The stomach's going to be sitting right up here. And it's located right, the head of the pancreas is actually located right along the descending part of the duodenum. So here we have the descending duodenum. The head of the pancreas actually comes right up against the descending part of the duodenum. And if I were to, I'm going to erase those names now, just so I can draw the duct system within it. Most of the pancreatic tissue is involved in producing a fluid called pancreatic juice. So we got specialized exocrine glandular epithelial cells here. They're producing a fluid. And that pancreatic juice drains through two major ducts. The main one that comes through the whole length of the pancreas is called the pancreatic duct, and then branching off the pancreatic duct and coming out of the pancreas at a slightly different location is the accessory pancreatic duct. So let's just write those names down here. Pancreatic duct, and then we've got accessory pancreatic duct. I'm going to write PD for short, pancreatic duct, accessory pancreatic duct, and that is this one. Now obviously, since these two ducts are connected within the substance of the pancreas, pancreatic juice can exit the pancreas through either duct and actually get into the lumen of the duodenum. In addition to producing pancreatic juice, which is important in the digestive process, and we'll see what all the enzymes that it contains here in just a moment, the pancreatic tissue produces a couple of hormones. There's some isolated groups of cells that are scattered throughout the pancreas called pancreatic islets. They used to be called islets of Langerhans years ago. And those islets actually produce the hormones insulin and glucagon. So we think of the pancreas as a mixed endocrine-exocrine type glandular structure, producing as an exocrine product pancreatic juice, and as an endocrine product insulin and glucagon, two important hormones that are involved primarily in glucose metabolism. The pancreas can become inflamed quite easily, and acute pancreatitis is just really no joke. It's quite painful, and it can be life-threatening, in fact. Obviously, because it has so many important functions, you can't remove the whole pancreas, basically, but some parts of it can be removed, and you may have heard of some of your friends or relatives that have had pancreatic cancer. Quite often it spreads so rapidly and is so malignant that those individuals have a short lifespan, even if they have surgical intervention. But in some cases, in some types of pancreatic cancer, the individuals can survive it and beat it. At any rate, that's kind of the story on the pancreas. Let's look at a couple of graphics that will summarize what we just talked about. The parts of the pancreas in this graphic are head, body, and tail, and then pancreatic duct and accessory pancreatic duct. Now, I didn't mention it when we were talking about it, but the pancreatic duct does join the bile duct, and we'll mention that some more when we talk about the biliary duct system in just a bit. So just those three parts from a regional standpoint, the two ducts, and that's the story from a gross anatomy standpoint. Any pancreatic questions before we move on? Oh, well, I want to look at this second graphic, though. Let's take a look at this next graphic that talks about the enzymes that are present in pancreatic juice. Largest and most important group of hydrolytic enzymes in the digestive process. Look what we have. 1, 2, 3, 4 enzymes that hydrolyze proteins. Trypsin, chymotrypsin, elastase, carboxypeptidase. A couple of enzymes that break down lipid materials, phospholipase and lipase itself. A very strong amylase. Now, we mentioned amylase in regard to saliva, but that particular amylase is a very weak one. This is a very strong one. And so this is very important in the breakdown of starch. Got cholesterol esterase, which will break down cholesterol. Ribonuclease and deoxyribonuclease will break down RNA and DNA, respectively. So you can see how important pancreatic juice is to the digestive process. In the small intestinal lumen, then we have pancreatic juice with all of these enzymes that's present, helping break down foodstuffs. And we also have some enzymes that the small intestinal cells themselves produce that are important in the hydrolysis of certain materials. So that's kind of the story on the pancreas. Let's go to the liver. I want to bring a liver up here. In this plastic model, we're looking down on the top side of the liver. That would be the portion that would be adjacent to the diaphragm. And this blue structure right here actually is the inferior vena cava, which is the main venous channel that's returning systemic blood back up toward the heart. This liver is the largest internal organ. It's also the largest gland in the body. It's the second largest body organ. Anybody know what the largest body organ is? We're talking by weight. Eva?
The skin?
Skin. Skin's the largest organ by weight, but the liver is the second largest, so it gets 2 out of 3 gold medals and a silver. That's at the Organ Olympics. Now, I'm going to go back to this, this overhead camera one more time, and let's look at the inferior side of the liver. So I've turned it over, and we're looking at the bottom side now, if you will. The liver is actually divided into 4 lobes, and the gallbladder here, which is the green structure in this location, kind of helps divide the parts of the liver up one from another, because the 4 lobes kind of blend into each other and are not cleanly separable. This portion right over here on this side of the gallbladder that I'm kind of covering up with my hand is the right lobe of the liver. And this portion over here that I'm covering is the left lobe. So left lobe and right lobe are the biggest 2 portions. This segment of liver right down here that has the gallbladder between it and the right lobe is the quadrate lobe. And this segment right up here, kind of around the inferior vena cava, is the caudate lobe. Let's look at those words on a graphic to make sure that you get the spelling of those down, particularly on left and right, huh? Left lobe, right lobe, caudate lobe, and then quadrate lobe. Those are the 4 lobes. Now, even though the liver has this gross lobation, this division into these 4 lobes, there is no clinical significance or physiological significance of that lobation. It's just the way the liver is. Now, what does the liver do? Well, what doesn't it do? It's an amazing organ. It has an exocrine product. What it considers to be a waste product comes out through the biliary duct system, of course, is bile. And that's an important digestive fluid in its own right. We'll talk about that a little bit more. But I want to kind of get down to the substructure of the liver so that we can understand how the liver does what it does, and then we'll kind of explain what it does in that process. So let's go to the overhead camera, and I want to draw a representation of a microscopic view of the liver. So what we'll do is we'll pretend like this is a little group of liver cells here, and here's a little group of liver cells here, and another little group of liver cells here, and so forth. The individual cells, I'm just going to kind of draw as these little black dots. Now, I'm not going to fill this all the way in. But the liver cells are kind of arranged in plates that are kind of 2 cells thick that radiate toward a central area. Each group of liver cells that are kind of physically arranged in the same manner, same thing would be here and the same thing here, each group is called a liver lobule or hepatic lobule. Let me write that down, hepatic lobule. That's just this group. So we got 1, 2, 3 hepatic lobules represented. This central, kind of a bullseye in the middle of the hepatic lobule is the central vein. And when blood comes into the liver, incoming blood, it actually comes in the adult through 2 vessels. Some blood comes in by way of what we call the hepatic artery, And that when it gets inside the liver, that hepatic artery breaks down into small branches that have little small muscular arteries that go all the way around each lobule in 5 or 6 different locations around each lobule. So let each one of those circles represent a little branch of the hepatic artery. There's a venous channel that comes into the liver as well, and that's called the hepatic portal vein. And when that hepatic portal vein gets into the liver, it breaks down into smaller venous channels. And finally, we get down into little tiny channels that accompany each one of these branches of that hepatic artery. So if we go back to the overhead camera, in addition to the little arterial channel, there would be a little venous channel. I'm just going to draw a little bigger circle here by each one of these and say that's the branch of the hepatic portal vein that's also located around each lobule. Finally, to complicate this picture even more, there's a little tiny biliary ductule that's located in each one of these locations, right along with the other 2 vascular structures. So what we end up with is kind of a trilogy, if you will, 3 little tiny tubular channels. And so what we're going to say is this set of 3 is just like every other set of 3, and that has a branch of the hepatic artery. I'm just going to write A period for artery. It has a branch of the hepatic portal vein. And it has a biliary ductule. Now, these 3 things, found in several locations around each lobule, each set is called a portal or a hepatic triad. So the name for that group is a portal or hepatic triad. Now, the blood that's coming into the liver by way of the hepatic portal vein, is that high oxygen blood or low oxygen blood? It's low oxygen because that blood has already gone through a first capillary bed out in the wall of the stomach or intestine or maybe within the pancreas or within the spleen. We'll go through that in just a minute. I want to kind of diagram that hepatic portal system, which we didn't do when we talked about the cardiovascular system. That's low oxygen blood. But that low oxygen blood that's coming in to the liver by way of these branches of that hepatic portal vein does contain metabolites, perhaps, that have been absorbed from the alimentary canal that the liver needs to act on. There are many materials that are absorbed that either need to be detoxified or metabolically modified by liver cells, and this is a way that that material can be taken to the liver for that purpose before those materials go into the rest of the body. Now, since that has low oxygen blood, the blood that's coming in by way of these branches of the hepatic artery is present in order to meet the oxygen requirements of the liver cells. So we've got both high oxygen blood and low oxygen blood coming into the liver. And then as the liver cells produce bile, that bile can be conducted out of the liver by way of these little biliary duct channels. Let's go back to that overhead camera one more time. The blood from both the little artery and the little vein flows down in between liver cells into this central vein. And those modified capillaries that it's flowing through in that area are called liver sinusoids. Let me just write that word up here, sinusoid. That's a modified capillary within the liver itself. So both the high oxygen and low oxygen blood flow down through the sinusoids. As that blood flows down through these sinusoids, the liver cells metabolically act on the metabolites within it. The materials are pulled out of the bloodstream, it's flowing down through there, and they liver cells detoxify or change things as necessary. And as they do these kinds of chemical transformations, the waste product that they produce as a result of that is called bile. So they do produce bile as a result of the things that they're doing to the ingested materials. Now the bile flows into some channels that are separate from the sinusoids. In between the liver cells, there's some little teeny tiny ductules that carry bile. They're actually called bile canaliculi. So we got sinusoids carrying blood, and we got bile canaliculi carrying bile, both of which are found within each lobule of the liver. Obviously, the blood is flowing centrally into the central veins of each lobule, and the bile is flowing peripherally to get out into that little biliary duct that was found within the portal triad at the outside edge of each lobule. Now, when a toxic material is ingested, like alcohol over time, the alcohol coming out of the bloodstream will actually begin to damage the liver cells, and it damages them in a characteristic pattern, because the ones that are located near the outer aspect of each lobule will be the first and most severely damaged by that alcohol. By the time that alcohol, or other toxin for that matter, gets further down the sinusoids, more metabolic transformations will have taken place, and so the toxicity will be reduced. So a chronic alcoholic will have the outer part of each liver lobule damaged by the continued exposure to alcohol, of those cells to alcohol. On the other hand, we take an individual that has heart failure, has congestive heart failure, and you get a backup of pressure into the central venous channels. The back pressure, let's go back to the overhead camera for just a minute. The back pressure in these central veins will be increased such that we expect to find damage around the central veins in, say, congestive heart failure. So a pathologist can take a look at a biopsy of the liver. In many cases, can make some important diagnostic decisions based on what he sees in that section. Yeah.
So in both of those instances, does the liver swell and get bigger?
Liver increases in size. Liver makes an abortive attempt in the case of cirrhosis from chronic alcoholism, for example, to remain functional, but finally so few cells, so much scar tissue develops within the liver, so few liver cells are functional that it can be obviously fatal, and is sometimes. Let's go back to the overhead camera one more time, and I want to just say this is a liver. Coming into the underneath side of it, then, we've got two different vessels carrying blood in. We mentioned those by name as the hepatic artery. It's actually, by the time it gets to the liver, it's called the proper hepatic artery. And we mentioned that it was high O2 blood, high in oxygen. And this over here is the hepatic portal vein. And of course that's low O2 blood, because this blood has already been through a capillary bed in other abdominal organs. After going through the liver, through those little tiny vessels that we just drew previously, the blood exits the liver through 3 or 4 hepatic veins, which actually are kind of buried within the liver tissue itself, and those empty into the inferior vena cava. So as blood leaves the liver, it goes out hepatic veins, not to be confused with hepatic portal vein. Hepatic portal vein carries blood into the liver, but these are hepatic veins which enter the inferior vena cava. I'm just going to write VC here. So I want to make sure you understand we've got 2 vessels in the adult carrying blood into the liver, 3 or 4 hepatic veins that drain the blood out of the liver, and that goes straight then into the inferior vena cava. And that blood then returns onto the heart. And of course the blood then goes systemically, and whatever metabolic modifications or detoxifications have been performed by the liver then are good for the whole body. What I do want to talk about for just a little bit, and we haven't talked about yet though, is the hepatic portal system, the pattern of blood flow that finally gets blood into this hepatic portal vein to go into the liver. So I want to go back to the overhead camera, and we've already talked about the cardiovascular system, but let's say this is the diaphragm right here, and coming through the diaphragm we've got the aorta, which comes down here and bifurcates into left and right common iliac arteries. So this would be the abdominal part of the aorta. Coming off, there's several branches that come off here, but coming off up here pretty high is a large branch of the abdominal aorta. It's called the celiac trunk. I'm going to write the name right here, trying to leave myself enough room to get all this on. Celiac trunk. What we say that if a vessel had the last name trunk, an artery had that last name, what would that mean? Starley, what would it mean?
Oh, it just means it's going to split really soon.
It's going to split really quick. The celiac trunk actually splits into 3 branches shortly after its origin, and one of these goes directly to the liver, one goes to the spleen, and one goes to the stomach. Just a little bit down below that is another fairly large vessel that comes off and has a whole bunch of branches, but it's called the superior mesenteric artery. Superior mesenteric artery. I didn't leave myself quite enough room. It goes mainly to small intestine. Some other things, but the main thing is small intestine. Finally, there's a much smaller branch that comes off down here. It's called the inferior mesenteric artery. I'm going to write M-E-S period for mesenteric and A period for artery. Now, the inferior mesenteric artery goes primarily to large intestine. I-N-T-E-S for intestine, okay? I'm going to leave the liver out of this. I'm going to circle these. After going through what we call a first set of capillaries in these organs, spleen, stomach, small intestine, large intestine, and oh, by the way, pancreas is in there and so forth. But after going through a first set of capillaries, in these organs, that blood is collected into some venous channels that coalesce to form the hepatic portal vein. So that's where that hepatic portal vein is derived. Then this blood goes where? To the liver. We just talked about that. And within the liver, within the substance of the liver, it goes down through those sinusoidal spaces, which are considered to be a second set of capillaries. And then the blood goes through into the central veins of the various hepatic lobules and drains out of the liver through those hepatic veins. So the point that I'm trying to make is this is called the hepatic portal system. Remember, a portal system was a situation where blood went through 2 sets of capillaries in 2 different locations before that blood returns to the heart. And we mentioned previously that there are only 2 portal systems in the body, one of them between the hypothalamus and the pituitary gland that was called the hypothalamo-hypophyseal portal system, and it was involved in transport of certain hormones. This is called the hepatic portal system, and it seems to exist in order to allow the liver to have the first metabolic exposure to ingested materials before those things go systemic into the rest of the body. Anyway, that's kind of a schematic misrepresentation of the hepatic portal system. I want to go back to the overhead camera one more time on this. If we go up here, you notice I left the branch of the celiac trunk going to the liver out of the deal. That is going to be the hepatic artery that also comes down to the liver. So this vessel right up here that I pointed to and said liver, that's the hepatic artery. Now for you purists, this vessel going to the spleen is the splenic artery. This vessel going to the stomach is the left gastric artery. And these vessels going to the small intestine are primarily jejunal arteries, although there's a, pancreatico-duodenal artery and so forth and so on in there. At any rate, all we did was point to the organs that those branches went to. Any questions about what we mean by hepatic portal system? Adam?
The blood that comes out of the hepatic artery, is that to feed and nourish the liver itself?
Yes. It's primarily there to meet the oxygen requirements of the hepatocytes, the liver cells. Because remember, the other blood that's coming into the liver is low oxygen blood, and so that won't do. Now, obviously, there's a mix. So those go down through the same sinusoid. There's a mixing of the blood, but evidently there's enough oxygen there that the hepatocytes can pull out enough to meet their needs. Any other questions on that? Yeah, Bryce?
Now you said that the lymphatic system pulled lots of lipids out of the intestine, so does that just skip the liver and lots of fats don't go through the liver?
If a lipid material is absorbed through the wall of the intestine, it's obviously not going to go into a blood capillary, but go into the modified lymphatic capillaries. That would be called lacteals. I don't think we mentioned that before. But those lymphatic capillaries obviously are not going to end up in the liver. Those go into the larger lymphatic channels, which eventually dump into the two big, all these in the abdominal cavity actually are going to go into the thoracic duct. So all the lipids that are absorbed from the alimentary canal are going to end up in the thoracic duct, which is going to dump into the left brachiocephalic vein. And so the first major organ that those absorbed lipids are going to encounter would be the heart. It's kind of interesting, too. The heart can make pretty good usage of lipids for energy. And so the lipid materials do go straight to the heart without passing through the liver first. Any other questions? Seth?
Yeah, I was wondering if you could tell me how adept the liver is at repairing itself after being damaged by like alcoholism and even physical damage.
The liver has a fairly large regenerative capacity. You can actually remove a significant portion of the liver for whatever reason, and it's one of the few organs that will partially regenerate lost tissue. But in the case of chronic alcoholism, we simply overcome the ability. There's so much scar tissue laid down that the metabolites can't diffuse through that tissue to get to the hepatocytes that are regenerated. So it's an abortive attempt at best, and we certainly can't overwhelm the liver in its ability to regenerate. But it has a remarkable regenerative capacity. Adam?
In some, liver conditions, causes a kind of a jaundice, a yellowing throughout the body. What causes that?
Let's talk about the bilirubin cycle. One of the things that the liver does is actually, it's one of the organs that is involved in the breakdown of hemoglobin from, when we have red blood corpuscles that have been in the bloodstream for X number of months and they finally wear out and are ruptured when they pass through bone marrow sinusoids or splenic sinusoids or perhaps even the liver itself. When the hemoglobin is released, let's go to a graphic that illustrates this, Adam. If we go to this graphic right here, when the hemoglobin is released from a ruptured red blood corpuscle, the hemoglobin is broken down in the liver into heme and globin. This is the protein portion. The heme portion is the portion of the molecule that contains iron, and of course the iron is actually the oxygen-carrying material in the hemoglobin. Each hemoglobin molecule actually has 4 different proteins in it, 4 different protein strands, and each hemoglobin molecule has 4 hemes in it. Each heme can carry 1 molecule of oxygen. And so, at any rate, when the heme is broken down, the iron is taken out of it, and the resulting material after you remove the iron from the heme is called free bilirubin. Now, free bilirubin is not particularly soluble in blood, But that goes through the liver cells can actually be converted into what we call conjugated bilirubin, which is pretty soluble. This imparts a yellowish material or yellowish tint to the skin. And the conjugated bilirubin, some of it is actually secreted into the bile where it goes down into the small intestine. Bacteria in the small intestine will convert some of that conjugated bilirubin to a yellowish pigment called urobilinogen. And that is the pigment that makes feces kind of a normal yellowish brown color. Some of that urobilinogen, however, can be reabsorbed into the bloodstream from the lumen of the small intestine. It's bacteria that actually form this material from the conjugated bilirubin. When this is reabsorbed, then it is filtered by the kidney and enters the urine that's formed and gives the urine its yellow color. So this yellow color right here, or urobilinogen, is responsible for the yellowish color in urine. So anyway, that's kind of the cycle of events involving bilirubin. When you have a large amount of bilirubin in the bloodstream, you can have a yellowish color that we say that individual is jaundiced. And it's not a disease syndrome in and of itself, but it can be indicative of liver problems or some other kinds of things as well. Other questions? Starley?
Sorry, how does sunlight play into that?
Like in newborn infants, sometimes they have a liver that's not doing everything that it's supposed to be doing, and sometimes exposure to some ultraviolet light somehow will facilitate that jaundice situation, but I'm not sure exactly what the pathoetiology of that is. Any other questions? All right, I want to pretend like we're going to do a transverse section through my abdomen right here. Okay? Texas Chainsaw Massacre, Mike Shively. Be a bloody mess, but we'll pretend like we can do it cleanly and neatly. And I want to go to the overhead camera and say, okay, that's my abdomen in transverse section. If I put a kidney right here and a kidney right here, does kind of look like Kermit, doesn't it, coming at you a little bit, but there's a serous membrane that lines the abdominal cavity, and I don't have colors here, so I can't show you different colors, but I'm just going to draw a black line. That serous membrane is called peritoneum, and it basically coats the outside of everything that's in there and the inside of the abdominal wall. When the peritoneum is just on the abdominal wall, like it is here, we call that parietal peritoneum. It's kind of like a saran wrap layer, if you will. And when that same stuff goes across a major organ like this kidney, that's called visceral peritoneum. The space in here is called the peritoneal cavity. Now, obviously, there's a lot of other organs in here. If we were to draw a section of intestine and another section of intestine, and we could put spleen and some other things, but I don't want to overcrowd this. What the peritoneum does is it comes down and surrounds each one of these organs and coats that organ on the outside. And so the peritoneal cavity itself is actually very small because there's so many things that are jammed in here. It's kind of a potential space. But when you have peritonitis, for example, you have inflammation of the peritoneal membrane. Can result from some kind of infectious process, and it's certainly a very serious condition. But if we go through here like this, I just want you to realize that this particular black line with this arrow that we've drawn would penetrate parietal peritoneum and then be visceral peritoneum and then visceral peritoneum, visceral peritoneum, visceral peritoneum, parietal peritoneum as it passed through this direction. And when you take a look in a surgical suite or even on a relatively fresh cadaver, this peritoneum is a very slick, shiny, forms a very slick, shiny surface. And of course you can get fluid that collects in here too in certain conditions, ascites or collection of fluid out in the peritoneal cavity. And have you seen the pictures of the, really heart-wrenching pictures several years ago of some of the starving African children? And they had this great big, they were skinny as rails, but they had this great big pot belly. And they had this collection of fluid. Basically what's happening in those cases is they're under the malnutrition state that they were in, their liver was unable to produce adequate albumins, and so the osmotic pressure of their blood dropped so low that they were leaking fluid into the peritoneal cavity. Real sad situation. But at any rate, I just kind of wanted to show you that relationship on this drawing so you'll know what we mean by peritoneum and peritoneal cavity, parietal peritoneum and visceral peritoneum. Starley?
I just had a question on that membrane that was actually going to the intestine where it was actually touching each other. Do you call that visceral?
Oh, let's go back to the overhead camera for just a second. What Starley's talking about is she's talking about this peritoneum that comes down, goes around a piece of intestine, goes back to the posterior body wall. What we have here is like 2 layers of saran wrap stuck together, and there's actually individual names for those. When it's on an organ, we call it visceral. When it's on the body wall, we call it parietal. But when we got the 2 layers stuck together, and there's a lot of these, those are all given special names. Like that comes down to the colon. If this piece of intestine were colon, it would be called mesocolon. If it comes down to a small intestine, it's called mesentery, and that sort of thing. It comes down to female reproductive system, it's called broad ligament. That's just, those are just the names that are given to those particular double folds, 2 layers stuck together. So there's actually about 20 of them by name.
So if you were going through it, would you say it was 1 or 2?
2
layers?
serous surfaces, but it looks like one. You can actually physically pull them apart in the lab, but they're stuck together. Like 2 pieces of saran wrap would be stuck together, yeah.